also going to put some materials. This is just to review and prepare for the exam. Review for the outlook and prepare for the exam. And if you think that this topic is interesting and some of your fellow students might be interested, we also have a summer school, and you can find it by search the internet maybe, but otherwise it's like this. In fact, our field is really, yeah. Okay, so we have we have talked about many things, and today we talk about history of vision, and Q&A for preparing for the exam is only one aspect. Another is Q&A on anything you like related to the future of the field, how it's going to be, and so on, okay? Or just technical things like, I want to ask some specific question here, there, things like that, okay? So, as I said, we have some summer school, which is a little bit of a different style because it's lots of biologists, psychologists, and physicists and computer scientists. It's quite a mixture. And you see that we have done all these, and so today is, review and history outlook. And so the idea is you are trying to maybe prepare for tomorrow as well as for your future. And so, what did I? No? Yeah. So, so this is for today. For example, one very practical question is, what is the exam tomorrow? Okay, so if you have done all these lectures, maybe at least watch some of the videos, try the quizzes, and do some of the exercises. Then the exam will be in between the quiz and exercise, roughly like that. Yeah, so it will be maybe some of them as easy as the quizzes, which is more or less keep your attention during the videos. And you may want to just practice on online courses and see, I can do the quiz or not. Yeah. And if you find these exercises useful, maybe there's some questions that you will find easy to answer. And I do understand you have a big workload. So if you think that, I haven't done maybe the full video, I haven't done. So the idea is if you have understood most of things, attended all the lectures, you are probably quite likely find it not too bad. But we do want to have a few questions to see whether you can do it. So if you are doing really well, you will have a chance to shine as well. Yeah. So the question is, do you like to have a chance to shine, or do you just only want to pass? You can plan accordingly. Okay, so let's go to the history. The history really goes very early. Yeah, this is 2,000 years ago. Or 2,000 years ago, ancient Greeks and stuff. So in fact, in early days, Aristotle had the idea that what part of the organ of your body does the thinking? He said the heart does the thinking, not the brain. Huh? So there's one theory says the brain does the thinking. Another theory says the heart does the thinking. How do you experimentally test which theory is correct? How? Anybody have an idea how to test which theory is correct? Do an experiment. What do you think? Which experiment would you do to test? Yeah? Fundamental science, yeah? Science is you have a theory, you have to test it. Yeah, please. I'm talking about experimental tests between those theories, not anything else. You are thinking about experiment to test those theories. Between which theory is correct, whether your brain does the thinking or your heart does thinking. Which experiment to do? Are you talking about experiment to test between which theory is correct? Okay, tell me the train, railway, and then... How does that experiment test whether it's the heart or the brain does the thinking? Yeah, and we also have in the history lots of other theories. The earth is flat is one theory. The earth is round, another theory. How to test it? Do you know an experiment testing between those two theories? Which experiment? What? Another place, okay. I see. Okay. Yeah, that could be an experiment. Another experiment, just sail to the west, you hit India. That's another experiment. Columbus did it, no? You say, oh, I have a prediction. If the earth is round, I can sail to the west and hit India. Yeah, and so there's lots of things. We can just think simple. So the idea is in science, we observe something, then we have an idea, we observe the star rises and we say, the earth is flat. Or we think, the earth is round, and you do experiment, yeah? And so in old days, in ancient Greek, we say, the heart does the thinking. A simple experiment is cut the heart out to see if the person continues thinking. If the person stops thinking, if you cut the heart out, maybe it tests the theory, yeah? But just because this prediction is confirmed doesn't mean the theory is correct, yeah? And so we can also cut the brain out, and this person also stops thinking, that theory is also confirmed. What can you do, yeah? So these are the things that we face when we... Anyway, in the old days, 2,000 years ago, people say how we see is we shoot. One is called extramission. That means we shoot light from our eye to you, and I shoot light to you, and then that's how I see you. Another theory is you shoot light into my eye. That's how I see. So you can imagine people have been wondering about how the brain works, how the vision works for many years. These are two different theories. Okay, one is called extramission, one emission theory, emission theory. And about 1,000 years ago, this doctor in the Arabic world doctors are great in already knowing the brain, and she knows, okay, there's a retina, and then somehow go to the brain. And they must be doing a lot of vision, okay? He wrote a lot of these interesting books, like a book of optics. It's interesting. A lot of great people write books of optics. Isaac Newton also has a book of optics, and Herman van Helmholtz also have book of optics. It's as if great scientists like to book of optics. But there was a great confusion. How can light enter the eye in a very smeared way, but we see things very clearly? I see your face very clearly. But the light enters the eye, the eye has a pupil. It must be many images. It's all very smeared up. How can you see? But how come we see very clearly? It must be the working of the brain. By then, it's acknowledged the brain is doing the thinking. The brain cleans up the image, and then I see you very clearly. That was the idea. But then how? Don't know. Until, as I said, that Johannes Kepler, in his trying to understand the celestial bodies moving around in the sky, understand the working, how the telescope works, and then, it must be how the retina works too, how the eye works. There must be some lenses, just like a telescope, and this lens must be having an adjustable power, okay? And so that's great, and that's why Isaac Newton started to also stand on his shoulder, trying to understand through the optics. Indeed, this is a lens like the telescope. This is a lens like our eyes, and indeed it will bend the light. And in doing that, he discovered, wow, it not only bends the light has all different colors. So that's another discovery. All of these are discoveries and theories and discoveries. And discovery is experimental fact. And throughout this course, we have had some discoveries by a lot of demos. I make you look at it. Do you see this? Do you see that? All these are experiments. And we need to get used to the idea. It is actually experiment. You should treat it as an experiment. Maybe not as a clean experiment as you do in CERN, high-energy physics. But when Galileo dropped 2 balls from a Pisa Tower, it's the same kind of experiment. Not as clean as in CERN, but nevertheless a great experiment, okay? So when I make you, do you see this? Do you see that? Do you see this illusion? Do you not see this illusion? These are all, do you see that this thing attracts your attention? That thing doesn't attract your attention. It's all experiments, yeah? It's just experiment measured in a very coarse way, not in an accurate way. If you really want to be accurate, then, it's just quantitative difference, but qualitatively, it's the same experiment, okay? Or maybe experiment measured in a way that you just sit there as a student, you're already thinking about this, and this experiment, you are biased because you already heard what I said, so the measurement result will have a systematic error compared to somebody who haven't heard my lecture and come to see, So a lot of these physics experiment about errors, systematic errors, unsystematic errors, these are all applied, yeah? So he did this experiment as well as poking his eyes. Poking his eye is also experiment, yeah? So all these kind of things. And then there's an Italian doctor who ended up being actually quite poor, but died of poverty. But nevertheless, his name is Gennari. And people who think the brain does the thinking. The whole brain is a piece of homogeneous thing, does the thinking. But he noticed that somehow the back of the brain have these kind of stripes, which is lacking in the front of the brain. The back of the brain is actually primary visual cortex. But of course, at that time, people did not know. He said, oh, back of the brain must be special. In what sense is special? Nobody knows. And so this is about, 1782. So then Thomas Young, who you probably know from physics because he did the double slit experiment to show that light is a wave, light is not a particle. And if you think light is a wave, since we see by the light, then how does vision work? So he realized that if light is a wave, it's not, you don't necessarily need all these different colors. Maybe only 3 is enough. How did he guess 3? Can we have an idea how did he guess 3? Just for the sake of thinking. How did he guess it's 3, not 4, 5, 2? How? What? Did he know that? Now that you know that, at the time did he know that? How did he know 3? This is used theoretically because after he developed, yeah, before he developed. How did he know 3? Yeah. How did he guess from nature? Which part of nature make him guess 3? Yeah. Uh-huh. I see. But here you can see the different colors, 7 colors. Why did he say 3, not 7? Yeah, so that's the question. People can see the difference between cyan and blue, cyan and green, green and yellow, yellow and red. So So the question is, how did he? Can you imagine? Yeah. What? A bit louder, please. Yeah, we perceive, in which way? We perceive 7 colors. Which way? The wavelength is 400 to 700 nanometers. From 400 to 700, how did he guess 3? Okay, so, in old days, scientists are craftsmen. They do all kinds of stuff. Just earlier, we have these Renaissance people. In Renaissance, people do paintings, yeah? Da Vinci does paintings. So imagine you're Da Vinci. Would you guess 3 too? No? Why not? Yeah, how many colors do you need to mix before you paint? 3. Okay, so if you're a painter, say, wow, actually I only need 3 tubes. I can't mix any color. Yeah, and so these kind of things can make you guess. I don't know how Thomas Young guessed it. I'm just trying to reflect. How did he say 3? Just out of nothing, out of a thin air. So everyday life, when you watch things, this is all. You can watch it casually. You can watch reflectively when you do painting. In old days, there's no PhD programs. Yeah, you just go Da Vinci, a poor boy. Go be an apprentice with a big painter. His first job is to grind up little things, make paints. That's his job. And then he ended up painting better than his masters. Yeah. So by doing that, maybe he's wondering what little stones I have to chop into pieces and make paints. And then, you're doing craftsmanship. Yeah. And then you are a scientist. So sometimes when you do cooking, you are a scientist too. You say, how do I do cooking? Heat it up, put some salt. You can cook the normal way, or you can cook like a scientist. Yeah. So all of these kind of things. Anyway, so he says 3. But without any experimental, further study, that's it. And then comes the great Maxwell, James Maxwell, who, as a little boy, playing with this spinning tops with colors mixed up. And then he said, let me think of a way to test that 3. So he actually did experimental measurement. And who did he measure on? He was just using himself as just like you use yourself to see illusion. He used himself, used his wife, used one of his students. He thinks that student is colorblind. It's just these casual experiments. But he did it so precise, so he actually measured these things before biologists even discover there's 3 cones in the eyes. Okay, so amazing. And whatever he measured to this day is still correct. Yeah? Just like you say, I measured this table is 6 meters, and then later on people say, actually 6.01 meters. Okay, maybe a little bit, more precise, but more or less he did it all correct. And this way maybe paved the way that Maxwell interested in light. By being interested in light later on, he wanted to follow Faraday. Faraday was a little boy without education and then became a great scientist, also doing all kinds of craftsmanship, yeah? And then Maxwell just looked at all this and then, electricity, magnetism, and then, wow, it is light. You wonder if he did not do this color vision, whether he will realize it's light. But anyway, vision science is great, yeah? You make great physicists out of vision science. This is just for fun, yeah? I love to look at history, and I also just love to share stories with you. It's a lot of fun. I like it when people smile when they, yeah, good. Okay. And so he did it for a few years, and done. And he started to electricity, magnetism, and so forth. And then he's a great doctor in Germany, yeah, Hermann Ludwig van Helmholtz. He's a great admirer of Maxwell. That's my hypothesis. I'm not sure. I never asked him, are you a great admirer of Maxwell? But anyway, he then simultaneously or maybe subsequently really did this three-color theory to a more precise degree, or maybe more communicated to the public. And he is also a leader of science in Germany, and he's also a doctor. He's an optometrist. Yeah, interesting. A lot of scientists are doctors in old days, so he just invented an optometroscope to look into the eyes. Amazing. And so people usually associate three-color theory with Thomas Young and Van Helmholtz, not as much with with the person who actually did a lot of it. Yeah, but that's history. Now, why do this is complete just to share a story with you. I love it. I say, why do I think that he is an admirer of Maxwell? Because Maxwell left and he did it even more on color. And then Maxwell died. Everybody got to die. But Maxwell died kind of early, yeah? Early such that he has this great Maxwell equation written, and that's it. Nobody tested it. And people think it's crazy. How could it be, electricity and magnetism, and suddenly there's waves and waves light. Nobody knows now. Who believed it? Very few people believed it. But if you are Maxwell's admirer, you would believe it, yeah? And one of the admirers is Helmholtz. And he's a leader of science in Germany, so he has his postdoc, junior faculty member. And so he asked one of the junior faculty members, whose name is Hertz. Guess who's that? Hertz. Now we say frequency Hertz. That's the Hertz. He said, oh, why don't you see if electricity and magnetism does create waves? Huh? And then, if you really admire, so this is why I don't know. But anyway, that's history, what happened. Hertz then tested by the short circuit of a thing. And when you have a short circuit, you have a spark. And then he measured a spark propagating in another. And that sparking measured in a distance without any electricity wire was the first confirmation that electricity and magnetism create waves. And then since then you have Marconi, another Italian guy, who then, you have the Marconi telegram and the modern era of electricity, magnetism, radio waves, everything comes out. Fantastic. Just some side story. Okay? And then people start to look at the corpse of the brain, and then sometimes with electricity you can do lots of things, such as, electricity, now people know electricity, yeah? You can stick electricity and trying to shock you, and you got shocked, and all these kind of things. So you can have a brain, and you give a little bit probe, and get shocked, and you find this animal twitch the arm, and say, if I shock this part of the brain, the arm gets twitched. Then the natural question is which part of the brain is not just twitching the arm, and it can make you see. And so people study, people have brain strokes and say, those people after stroke, they can't see. And then when they die, you can open the brain, which part of the brain is damaged. That's one way of noticing that, the back of the brain, kind of back of the brain. So that's called the posterior cortex. And then exactly where is not quite nailed down until this German doctor in Berlin. He said, it's actually really in the back. It's this part near the middle. And there's an English doctor who says otherwise. They said, not there, it's a little bit there. So they keep on fighting. Now, at that time, English was very kind of top of the world for science, because you have Royal Society, Royal Society for Scientific, stuff. And that Germany is also kind of getting more and more into this, especially with Helmholtz and so on. So this is kind of a contemporary of Helmholtz. Another doctor, and he said it's actually, by then they start to do experiment with monkeys, so it goes from humans and corpses and stroke patients to doing experiment with monkeys. How about you cut this bit out, then what? Cut that bit out, what? And so he says in the back of the brain. And then he's a Japanese doctor. Interesting, Japanese doctor at that time publishing German. But there was a big invention. The big invention was a bullet that's fast and small. So there was a war, Japanese-Russian war, with these bullets. So sometimes new technology like telescope make us see the sky and the Kepler's law coming out. So this technology is not a telescope, but a bullet. And this bullet can hit you and you may not die, okay? Because it's small and fast. So it can hit into the brain and exit from the brain and the soldier survive. Okay, imagine the bullet is too slow, too big. Then the soldier die, you get nothing. But the soldier survive, you can see. The bullet enters here, so you can see the wound. And then the soldier says, oh, wow, I can't see if it's over there. And then the soldier says, I can't see if it's over here. And so you realize that different bits is for different part of the visual field. So you map which part of the brain, because you can, so he get this device trying to measure exactly. Sometimes the bullet even hit the shoulder. You see, the bullet enters here, exits here, hit the shoulder. So you can reconstruct exactly where did the bullet go and got the brain. And then realize that this is actually precisely which part of the primary visual cortex is for which part. And this, remember, we talk about is like a topologically preserved. It's called retinotopy. We talk about how a small part of the fovea is mapped to a big part of primary visual cortex, V1, and how a big part of the peripheral visual field mapped into a small part of V1. And that he worked out. But interestingly, and this is a lesson to us all, yeah? So, interestingly, I never heard of his name until a few years ago. I said, how come I didn't know this person? And after all, I have written a textbook. And writing this textbook means that I have to read lots of things. And how come I didn't read his stuff? Yeah? I'm ignorant. And the reason was that he published it without a proper figure to explain what he discovered. Okay? So next time when you do your PhD thesis, and your supervisor asks you, make the figure more clear. Yeah? It helps because you can discover a great thing and nobody understands it. Nobody knows until 100 years later. Then, so this is a figure done by other people. So nobody knew that he did it. Yeah? So you not only have to discover, you also have to explain. And one way to explain is to teach. Teaching is great. So thank you very much for Tadio for being a TA. I'm sure that make you discover even more. Thank you very much. Yeah, and so to teach is to explain, and to write a paper to readers and to make the reader understand you is to explain. Explain clearly. Yeah, so that's interesting. And then, so that was 1900. Yeah, 50 years later, there was the first experiment done with an electrode to a live animal and not a corpse, kind of in mammals. So that was a cat. You can stick an electrode into the retina, and the cat is alive, maybe anesthetized, but at least alive. That's how we discover that the retina has center surround receptive field. Okay, fantastic. That's a milestone because before previously it's a corpse, it's a stroke patient, it's soldier, hurt. You could not really measure individual neurons, and this is really fantastic, individual neuron in a live animal. That was done in 1953. Okay, so therefore you can see 2,000 years ago, then 1,000 years ago, then Kepler 1600. And then, Newton and Thomas Young 200 years ago, Maxwell 200 years ago, and so on. And then the doctors. And now this is less than 100 years ago. Huh? So it's got 2,000 years of evolution for science to advance. Yeah, a bit too slow, but good news is then it becomes very fast. So he did it in 1953, Fantastic. And there's 2 junior faculty, or actually maybe more like a junior scientist or maybe like a postdoc, visit his lab. And these 2 junior people is Hugo and Weasel. Oh, you do that on the retina. Let's stick electrode to the next stage. So they look at LGN, they look at V1. And that's how they discover these orientation selective V1 neurons. And that was 10 years later. You don't have to wait 1,000 years anymore. So fantastic. 10 years is within your lifetime. And that's fantastic. And then, as I mentioned that, imagine you are a student, a postdoc comes in and say, great, 10 years, you can discover so much. And maybe I will do V2, another 10 years. And you know, V1 is 25% of the whole brain, whole visual cortex. So 10 years, 25%. Another 10 years, another 25%. And then you only need 40 years, then you understand the whole vision, huh? Well, 1960, now it's 2025. We already have 60 years past. So something is not quite right. Things got more difficult. And so this is the modern bit, and that's what I taught in this course. We talk about efficient coding. Efficient coding is more or less trying to understand what they did and what he did theoretically. Okay? I say they have the experimental data. Can we understand it theoretically? And can we predict slightly more that they haven't measured? That would be nice. That's efficient coding. And then, why is it that progress become slower? So you need to ask a new question. That's what I mean by modern vision science. And one of the modern vision science is to say, what is V1 doing? You know, but you can say, didn't they already tell you what is V1 doing? They tell you this neuron is orientation tuned. So why do you need to ask more, what is V1 doing? So the idea is, they already know what is retina doing, right? Well, retina, since Kepler says, well, it's just focusing light, form an image. And there's 3 cones in it, and now you all know all this. And people knew what retina is doing. Retina is a camera. The camera did a little bit more processing, that's it. Retina just samples the input and do a little bit more efficient coding. It's just sipped the, at that time, they didn't quite think of efficient coding, but more or less that's good enough, okay? You think about the camera with a little bit of processing. If you know what retina is doing, it's easier as a scientist to go to the next stage. That, okay, now given retina is doing this, what is V1 doing, yeah? And this may be the reason why it took them only 10 years. And so in this sense, people might think, okay, now we know V1 is doing this, it's ready to do V2. But maybe not. Why not? Any idea? So imagine you say, oh, I know how the hydrogen works. Huh? Hydrogen is an atom. You know, has a lot of, and I also know how oxygen works. Great, oxygen is another atom with some electron going around it. Hydrogen has only 1 electron, and so on and so forth. Perfect. You also know how carbon works. Yeah, carbon is yet another atom. So it's like, oh, I know how this neuron works. It prefers this orientation. I know how this neuron works. Prefers this orientation. I know how this neuron works. Prefers this orientation. But if you put hydrogen, oxygen, and carbon together, do you know what happens? Do you know how glucose is formed? Yeah? Glucose is the sugar we eat. So just because you know hydrogen, oxygen, and carbon doesn't mean you know sugar. The one that gives you energy is actually made of these 3 atoms, yeah? So in a sense that it's a big leap from knowing the atoms to the molecule. And the molecule can give you energy. That's a big leap. Without that big leap, you kind of not know. And so this is where, what does V1 do is we not only need to know the neurons. If we want to know intelligence, how vision works, you need to say, can V1 recognize my grandmother? No? Or can V1 do something beyond just a cell? Yeah? And so one of the things is, V1, move your eyes. Moving your eye is like, I walk around. This is a motor output, yeah? Like I grasp something, I take it up, pick it up. It's a motor output. What is the brain trying to do? Brain is taking signals in and do an action, okay? And so therefore this is, by knowing that helps us to know what's next. Because you say, why does the V1 want to move your eyes? Well, we move your eyes because it has to select a tiny bit of information. Well, if it selects a tiny bit of information, why tiny bit? I cannot recognize your face, only tiny bit. But somehow, maybe indeed tiny bit, and you realize that people do not see the gorilla walking around when people playing basketball. Wow, indeed, we are blind. And so therefore, we must understand vision despite this tiny bit of information. And so that's the framework. And this framework needs to be tested, okay? And so one of those things is, in science, we always need to test in the spirit of James Maxwell. So he not only have this theory coming from Thomas Young, 3 colors, he tested it with himself, with his wife, with his students, and then he said colorblind is what? And he got it, and prediction, everything tested, completely quantitative. So this is the idea, and we need to bring this tradition to vision science. And that's why I encourage physicists come to vision science and do it in a more that, because any science cannot be just doing experiment, okay? You need to also do theory. So these are great experiments, but experiments alone may be a little bit handicapped. We need great theory. And then the theory has to go back to experiments. Sometimes it's not always possible as one person who does who does both science and theory and experiments. Historically, if you look at great scientists, they always do both. Yeah? Maxwell, Newton, Galileo, they always do. But in modern science, sometimes maybe you cannot afford. Yeah, if you build up a big detector for particle physics, maybe you don't have enough time to think like Einstein. Difficult. Yeah. So here we are in Italy. Another great Italian physicist is Enrico Fermi. And Enrico Fermi is often referred to as the last great physicist who does both theory and experiment. Yeah. And so, good news, neuroscience, you can still do both. Okay? You can do theory and experiment. And we want to do it in a way that's as precise as James Maxwell, hopefully. So that's the idea. And then, by the way, it turns out that this particular theory can also be applied to many animals, except central and peripheral vision. It becomes central and peripheral sense. It may not be vision. It may be smell, it may be touch. But this is in search for general principles. I'm not going to dig into details. If you do come in theory, you may want to say, is there 10 theory or just one theory? We don't know. To understand the brain, do we have a grand unified theory or do we have 10 theories? But we need to find out, okay? So here is an attempt to unify sensory processing, whether the sense is vision or hearing or touching or smelling, and whether the animal is a human or monkey or cat or so. Any question? Okay. And here, I'm from Max Planck Institute, so let me advocate another physicist, Max Planck, whose Planck's law exactly matches the data, the 0 parameters. The data, could be more than 100 years after its birth. It's fantastic from Cobra telescope. And I want to say that we in vision science not as great as Max Planck, but we're getting there by having this theory experiment combination. And the reason we cannot be as great is these telescopes are so precise. They measure things so precise. We cannot measure human behavior as precise because each of these data points is only for a few trials. You cannot make human do too many trials because then this probability distribution will shift. Why would it shift? It's not a stable probability distribution because human will learn. So, if after 1,000 trials, human get faster. So you are not measuring a quantity that is a stable quantity. While these telescopes, they can measure, it's not exactly stable. The universe is actually changing, starting from Big Bang to current days. But you can wait a year to measure that. More or less, a year is not changing much, yeah? But in human behavior, a day is already a change. So therefore, it's a different kind of experiment. We need to think about how to measure the errors and so on and so forth. But the idea is once we find a way, then the science will move forward. And so it is actually a wrong concept to think that biology is messy. Biology cannot be messier than physics before Isaac Newton and before Galileo, okay? Galileo and Newton made physics neater, but before they made physics neat, it was as messy as biology, yeah? And so therefore, we need to make it neat by our own effort. And that's what's happening with the brain science. This is how the universe is changing. Universe starting from Big Bang, then get a bit more homogeneous, and then they get the telescope to so precise they find it's not so homogeneous, and so on and so forth. That's quite interesting. But these days you can have a telescope looking into the sky for a long time, collecting data, and then you can analyze data. Fantastic in all these great development. Yeah. And that's it. And let's have question and answers related to science, related to exam, whatever you want. Yeah? Okay. I hope I didn't make people go to sleep, but you are all kind of alert, so I don't think you went to sleep. Yeah? Or any feedback? Like, if you don't have question, you can give me feedback. Okay? So for example, we have a summer school. Well, actually, we also have PhD positions in case you are interested. Yeah? But even if it's not in my lab, you say, I want to do a PhD in somebody who's not doing vision, maybe doing something else. Would you tell me some? Maybe I can tell you, I know somebody, or maybe I don't. Okay, all kinds of stuff. Anything you want to ask. Okay? Or maybe you're not interested, but you know somebody else who is looking for this information. They cannot find it. You can also ask. Yeah? Okay. Mm-hmm. Mm-hmm. Yeah, it's very much studied, and we do not have an answer. In fact, we are considering to have a PhD project in this exact topic. And we have doctors nearby who happen to study schizophrenia. And so we can have these patients But, you probably seen this movie, The Beautiful Mind, and John Nash is having schizophrenia, great mathematician. Yeah? And so, yes, this is a very active area of studies. And in particular, they hallucinate not only vision, but also other things. But yeah, vision may be one window of looking at it. Yes, there's various theories, and so far it's lacking very solid, agreed upon one. Different people have different opinions so far. Some are more agreeing with data than others, but to say that people feel, oh, now we understand, not yet. So it's an active field of study. Yeah. Okay. Yeah. Mm-hmm. Okay. Mm-hmm. Okay. Okay. So, uh, for instance, if you're Eskimo, you have different shades of white for the eyes, so you actually have vocabulary for them. But actually, so far, I haven't found a solid evidence that they can distinguish different shades better than other people. The reason is underlying the red, green, blue are genetically there. These tuning curves are there. Remember, we have these tuning curves for different wavelengths of light, 400 nanometer for the blue and 700, okay? And our visible light is, the blue curve, the green curve, and the red curve. They are fixed, yeah? It's not as if different tribes, they are the same species, they are human. With this, we already see how the color discrimination, remember? This is the homework. And by the way, the last homework is color discrimination. You should also try to do. Anybody has downloaded that homework and done it? Anybody? No? Okay, sorry, Tade, I forgot to remind them. But it's useful to do that homework for the decoding. But we taught about it, remember? That how you can discriminate this. It's the same. It's repeated in different labs. And unless this human is a different species, but I don't believe they should be the same. Yeah. Of course, you can also be colorblind, and colorblind is by losing one of these cones, and then you will be poor. But I haven't heard anything more than that. Yeah. But vocabulary can be more richer on the shade of eyes and so on. Yeah. Good. Okay. Yeah.
Yes, I was discussing with one friend, and what happens when someone has just one eye?
Yeah.
The consequences are in the coding because you lost deep of the image.
That's
What
right.
we will most obtain in the equations is this.
Yeah. Mm-hmm. Yeah, yeah. And by the way, I forgot to ask another question, whether it's related to saliency. Saliency is to have you shift the gaze. Once you shift the gaze, saliency is useless. So if you say, oh, this shade of red and that shade of red are different, the gaze is already there, so it's not related to. By then, it's decoding with the information you already selected. And hopefully you select all these relevant information already. Then, yeah. So back to your question, yeah? If you don't have one eye, it's a little bit like if you don't have one cone. So you're a little bit blind in color, but that doesn't matter. You can watch black and white movie. You can still tell the whole story. So color is adding a little bit more, but not too much more. So adding another eye or not having another eye is not critical. Yeah, you can still show depths by just like, you can still see depths. You say this is in front of my hand because it's blocking the view of my hand. So the blockage, and also this is big, this is small, and by size. A lot of these will give you depth information. They are redundant information. But having two eyes with disparity adds a little bit of advantage. And this advantage is quite subtle. So people, you can go to clinic and test whether you're colorblind or not. But most of you have not tested whether you are stereoblind, right? Okay? Because it almost gives you no handicap. Because you can see depths by other cues. However, if you want to do fast sports, just want to do kick football and play basketball and ping pong, then you got to be really fast. You just have additional with two eyes. You can be faster. Okay, just imagine a ping pong ball shooting towards you. Okay, you say, the ping pong ball is small, and then get bigger, 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 and then you try to estimate the distance. But with your two eyes, you instantly can estimate the distance. Then you can already start hitting the ping pong ball. All you need is 1 millisecond faster than your opponent, then you win. Yeah? And so that's where, so sometimes people say, I'm not very good at playing basketball. I must have bad muscle. I have poor coordination. Well, it turns out that they were stereoblind. So I actually, in my experiment, everybody come in, I give them a free test of stereo vision. And I say, turns out about 10% of people are still blind, or depending on how blind it is. And this person said, my God, no wonder I cannot play basketball and all these kind of things. Just because you're still blind. And nothing to do with his muscle or nothing. And so that's what it is. However, you can play slow sports, such as playing chess, other things to do, no problem. Yeah. Good. Okay. Mm-hmm. Yeah, so some animals move their head this way. Yeah? That's it. You can have one eye, and that's also good. This is called motion parallax. And sometimes you can have a tiger hiding in the bush, you cannot see the tiger. But if you have two eyes, you can see a tiger. So a lot of animals camouflage. But with two eyes, you see the steps jumping out. Yeah. So for instance, we have this experiment with one bar in the one eye and the other bar in the other eye. They pop up. What's the use of it? Actually, they are used for these, animals not moving, so there's no motion for you. And they're static. And you can see. They immediately, they're salient, they catch your attention. It's called figure ground segmentation. Okay, so doing the homework will help you. You will find, I can answer that question because I did homework. Doing a little bit about, doing some of the quizzes in these, you can just say, this quiz I can do. Now I'm safe. That quiz I do not know, then watch the video just in front of it. Okay, that will help you. Things like that. You can have a little bit of feeling. Okay. Any other question? Okay. And those who ask questions are helping everybody. Some people are so shy they don't ask. So, yeah, please. Yes, that's very good. Paper and pencil, no internet, no mobile phone, no laptop. Yeah. Does not involve coding, but it might involve a little bit of calculation. But this calculation you can calculate by your head or you can calculate by doing a little bit. Yeah. Some multiple choice, some mostly multiple choice, but they will be slightly more difficult than the quiz questions. Yeah. I will give a shade of easy and to more difficult one. Yeah. So you're curious about human behavior, and the next sentence is? Yeah. So mostly I do it on adults. But if you have a scientific question that matters, that is on their age, then you probably should do the younger children. But actually, the quickest change in things are between 0 and 1 year, and those children are having a lot more difficulty to. But once you reach 1 year, you're almost adult. So therefore, it's no point to go to the children. And there's some little subtle change. The most interesting change is in the first year, or maybe first 2 years. Yeah. That's right, that's right. And this is one of the biggest challenges in doing experiments. It's not that the children could not do it, but maybe you just did not tell children what to do, and child is not paying attention to you when you tell the children. So for instance, do you see the disc in front or behind? And you all understood what I mean. You can say it's in front, behind. What if the child is not, and the child, And so these kind of things will induce lots of errors. And these errors, so doing, it's like measuring this table. You don't care whether the table understands me or not. I just measure the table. But in psychology experiment, you actually care that what you tell the subject to do, did the subject understand? Yeah, so you kind of need to do these kind of, if the subject do not understand, the error is huge. You have systematic errors. And this causes a lot of trouble in psychology experiments, such that you probably heard that the psychology experiment do not replicate. And such that in, Richard Feynman used to say psychology is soft science. And well, it's just that the way experiments were done was soft science. It should be done in the hard science, rocket science manner. And that's what we need, okay? Because it's a natural phenomenon. Of course, we do find some obstacles because we are studying our own brain. We may be not very objective. Measuring this table, I can be objectively measure the table. What if I need to measure my IQ? I may be upset if my IQ is too low, subjectively measure, yeah. And also we think we're not blind. Because we think we're not blind, we define the vision problem as no blind. And that's a subjective error we made in defining a problem. When you define a problem the wrong way, then it will have more difficulty. Those kind of difficulties are very special to brain science. Yeah. So sometimes when you measure children, the children data is different. Then we call that, there's an experimental confound. Is that really true the children is different? Or the experimentalist is not a solid scientist? Which one it is? Well, you need to find a way to, yeah. So, and if you want to measure children who are young, who cannot speak, they say, you see this in front or behind? The child cannot speak. But that's the same when you do experiment with the monkeys. You ask the monkey to tell you. Monkey could not speak. So you have to do some tricks of, those are very challenging, but they can be very interesting. You solve this challenge to get to very interesting data. Yeah. So, yes? Yeah. Previous experiment results are the foundations. You have to, otherwise it's kind of building sandcastle in the thin air. Of course, previous results, they were, messy, yeah? So for example, somebody did an experiment without knowing that you are going to have a theory explaining it. They didn't know, they did not anticipate that you are going to have a theory. So they may have done this experiment for a completely different purpose, yeah? But this is exactly what you need. And so as a theorist, you have to go find these is one of the biggest challenge. Yeah, a good piece of data and poor piece of data, they're buried in a pile. They're mixing the gold with rubbish. So you have to go dig the gold out of the rubbish. That's one of the biggest challenge. And then you have the theory of finally dig the gold out of rubbish. You say, now I guess the earth is round, the earth is not flat. Then you say, now what experiment can I do? Okay, you can sell the world, find India, you can go, measure. And then you say, nobody has sold the world, then better I sell it too. And so a lot of scientists will have to do that things. So you start from previous data. And it would be nice then it's not like every piece of data is already done. Then you have some piece of data that's not done. Then it's a great ground for testing your theory because you have no idea whether your prediction will meet the experimental data well or not. Yeah, that's a good chance to test the theory. Okay, yeah. That's the thinking, yeah. That's right. Yeah. Yeah, yeah. So, of course, these days we know the heart is just the energy, battery, supplying. But at the time, indeed, you can do both experiment. Cut the heart out, this person stops thinking. Cut the brain out, the person stops thinking. The both theory are confirmed, but two theories are contradictory. So you have to do another experiment. You say, how about I cut the heart out, but I give artificial heart. Keep on pumping the blood to the brain. The person still think. Then you say, it's the brain, yeah? Something like that. Yeah. And then you say, this artificial heart, all it does is pumping energy. That's it, nothing more. Then it's good. Yeah, good. Okay. Mm-hmm. Yeah. Computer vision is fantastic, but it's engineering. Yeah. So it's like, if you understand Newton's law, you can build airplanes. Yeah. But that doesn't mean you will build airplanes, or it means that, I want to build airplanes. So it's everybody's interest. Depending on, do you want to really understand nature? Understanding nature, it's discovery. You're not trying to invent anything. But you can invent the airplane. Okay, that's engineering. And so therefore, what we talk about today and all this, the brain science is natural science, while artificial intelligence is engineering. And they can benefit from our discovery in natural science, but they don't have to. Yeah, you say, I can build an airplane without understanding Newton's law. Yeah, if you can, maybe you've never studied physics, you can try to do an airplane. But maybe after you build an airplane, then you say, now I study physics and I understand Newton's law, then you can modify your airplane a bit better. That's the relationship. Yeah, definitely, because maybe there is just single discipline of intelligence, and I don't care whether it's a natural intelligence or artificial intelligence. I just study it, and then they build something that I've never seen in nature. Maybe that's fascinating. You may want to take advantage of it, yeah, and I believe so. Just when I said about all scientists used to be craftsmen, so engineers are craftsmen, yeah. And when Da Vinci trying to grind up the stones making paint, he was being a craftsman. And when you become a very good craftsman, he's also an engineer. He was also building all these shooting cannons, and he also dissect people's body, all kinds of stuff. So he's actually maybe doing both science and engineering. And a lot of people do both. Yeah. So in a sense, that if you really understand something, you can build it. But building can take a long time. And you say, maybe I just watch other people building, that's good enough. Or maybe you say, I actually want to build it. I don't want to do science anymore. That's also okay. Everybody's interest. Yeah. So that's the relationship. And if you want to ask me whether artificial intelligence will be better than natural intelligence, guess what will be my answer? What do you think will be my answer? Yes or no? Probably not. That's one answer. I would think yes. Why yes? My answer will be yes. Why do you think it's yes? Well, why do you think my answer is yes? From what I taught you 2 weeks, you can guess my answer should be yes. Why? Yes, they have more energy. They have no bottleneck. I wish I didn't ask you this question. Otherwise, I can put it onto the exam question. Yeah. What would be? Yeah, they have more energy. They have no bottleneck. They just imagine. Can you do? If I ask you to calculate 2319 times 4, 5, 6, 7. Is your calculate doing faster or you do faster? The calculate doing faster. So, calculate. So, to say artificial device is not superior than human is a little bit stupid because we already knew it was superior than human in some limited domain. But this limited domain can be expanded more. So, I believe it will be better. Yeah. Okay. No, they do not mimic. They only mimic V1. We do not understand what's beyond V1. And they pretend they're mimicking. They only mimic Hubble-Weasel's recessive field V1. They don't do the saliency map V1. So they only mimic very little. They do get results, just like you can build an airplane, you can fly, no problem. Yeah. And mm-hmm. Mm-hmm. Yeah, that's not a problem. Yeah. Yeah. Mm-hmm. 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 But CNN does not mimic the brain beyond V1. Okay. In fact, we do not understand the brain beyond V1 much for the moment. And we know that in industrial, you have these automations, and automation is already there before the neural networks. And all these automation already follow the line straight or bend. You can just tell it, but it tells it in a very brute force way. These days, with artificial intelligence, they try to tell it in a less brute force way. So by having all these parameters to fit your neural network is one way of brute forcing. And they can be very successful. You can have assembly lines on building your cars. A lot of assembly lines is automatic. Yeah, you have this, robot arm and doing all these kinds of things. They're completely brute force. They're very effective. Not a problem. So automated driving is yet one more. They're just a bit more flexible than your automated assembly line. Maybe I'm a little bit understatement to say a bit. You can say they are the extrapolation of more flexibility. Could one say it that way? Mm-hmm. Yeah. Mm-hmm. Yeah. Mm-hmm. Yeah. 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 Past 4 or 5 years, what is developing? RAG. Mm. I see. Okay. Yes, I'm sure they're doing great. Engineers, they can build airplanes much better than any natural flying birds. Yeah, so there's no doubt about it. Yeah, and you don't have to exactly mimic a bird's flying with feathers. Once you understand the fluid dynamics, you can build a steel wing and fly even more. That's no doubt. Yeah, and there's no doubt Google can do all these things, and because they have all the power, they have all, yeah, no, no doubt. So it's going to be very exciting. But maybe I'm a little bit biased. But on the other hand, you can say science and engineering always go hand in hand. Sometimes discovery comes before invention. Sometimes invention comes before discovery. So this is yet another field where you can have discovery and invention come hand in hand. It's very interesting. Mm-hmm. 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 Yeah, yeah. It's a very nice story indeed. Yeah, yeah. It's fantastic. Yeah. And so, for instance, I also want to add that, this is something I kind of briefly mentioned at the beginning of the course. Why do I teach these? Because in our field, theory that can be falsified are very limited. So I taught almost all except for James Maxwell's trichromacy theory. And so these kind of guidance hopefully can make us progress a bit more, make rocket science out of brain science, hopefully. Yeah. Okay. I like all this very, blue sky kind of talking. So, yeah. No specific question more on the exam? No? Okay, it's going to be 1.5 hours tomorrow at 2 o'clock after lunch. Yeah. And if no more question, we can say that's it. Okay. Thank you very much.